Let me start before just introducing Satish to point out that he and Nadia have been very helpful for the past several years, playing a very active role with CFAR, and Warren and I really appreciated that. Also, Satish is very involved in the pilot grant review process, chairing the basic HIV science review panel. And Jim Sorensen, who's here, has also played a role in chairing the clinical HIV study section. So, you know, our community is a really, really strong one, and that's they're good examples of that. Satish is Associate Professor of Medicine, or Laboratory Medicine at UCSF. Formerly has worked with Doug Richmond in San Diego, worked with Warner Green here at the Gladstone, is now at Vitalant, a research institute, formerly the Blood Systems Research Institute, and is very actively involved in the cure effort here, and he'll be talking about profiling and purging. Thanks, Satish.
Thanks, Paul, and thanks everybody. Yeah, so today I'm going to be talking to you guys about profiling and purging the latent HIV reservoir. And last night I was thinking, what would be the perfect back tattoo to represent this project? And this back tattoo instantly came to mind. So $5 for anybody in this room who can name this guy. Is that you? No. You wish that was me. Nobody in the room knows who this is? Mauricio, I'm looking at you. This is Henry Rollins, who is one of the greatest rock stars of all time. So Henry Rollins was the front man of Black Flag back in the day. I used to be a huge fan of Black Flag, but his search and destroy tattoo to me like perfectly represents this whole idea of trying to track down latently infected cells and destroy them. So I'm going to start by describing myself a little bit and the evolution of a CFAR mentee. So this goofy picture right here was actually me when I was in the CFAR mentorship program back when ice cream was a nickel a cone. And I think I had, this is when I was in Joe Wong's lab as a postdoc. I think I just started in the CFAR mentorship program. And I decided to take a mini sabbatical in Kerala in my family's ancestral village there. And I was looking back at the CFAR headshot, and it just seems like I have absolutely no cares. So then what happened was I got married and I had kids and I got a mortgage, and then I met Mike Bush and fell into a really bad crowd and started my own lab. Next thing you know, I'm looking like this, and I've just like given up. So this is what happens. This is the evolution of a CFAR mentee right here. But in all seriousness, what I really want to talk to you about was when I first came to UCSF, my main interest was in the evolutionary mechanisms supporting HIV persistence during untreated disease. And a lot of this had to do with the fact that I was mentored by Dr. Betty Korber at Los Alamos National Lab, who's one of the greatest HIV evolutionary biologists in the world, and also Doug Richmond. And when I came here for my postdoc, since I was working in Joe's lab, and Joe, as everybody knows, did seminal work to discover the latent HIV reservoir, my interests gradually shifted towards studying the mechanisms of HIV persistence in the setting of antiretroviral therapy. So I'm not really going to do much of a background intro here because I know you guys have already heard about latency a couple times today, but I'll just chart out the very basics in about 10 seconds here. So if we look at untreated infection, this is an overly simplistic schematic of what happens in the setting of untreated HIV disease. So we have an HIV-infected cell with thousands of them in the host. They're pumping out virus all day long. The virus encounters a population of susceptible uninfected cells. Those cells become newly infected, and then this process continues ad nauseum. But in the setting of treated HIV infection, now we essentially destroy this entire right side of the equation. So there's no more replication, or there might be a little bit of replication, but not an appreciable amount of ongoing replication. And so we delete this whole right side of the formula, but we're still left with all of these pre-existing infected cells. And a lot of these long-lived, latently infected cells are invisible to the immune system since they're not pumping out virus, and they're typically not displaying viral antigens at the cell surface. And from what we can gather, it seems like these cells can live for a very, very long time. So the goal of the HIV cure field in a nutshell, in a very simple nutshell, is to eliminate that pool of latently infected cells. And I think something else that we're interested in is this idea of a durable antiretroviral treatment-free remission or a functional cure, which is a slightly modified version of that goal. So instead of destroying every last instance of the virus in the host, maybe we can dramatically reduce this pool of latently infected cells and do something at the same time to potentiate the immune system so that it can control the residual virus. So here are key questions about the latent HIV reservoir that our lab is very interested in pursuing. The first one, how do latently infected cells differ from other cells? So in the schematic, the latently infected cells are the ones at the top. How do they look different from, I guess I could use technology. Oh, that's right. This is probably only works if I plug in my little USB. I'll just point. How do latently infected cells differ from uninfected cells and productively infected cells? What are the natural host immunologic determinants of HIV reservoir size? How can we estimate the size and also the anatomical distribution of the HIV reservoir? And perhaps most importantly, and something that I'm getting increasingly interested in, is how can we selectively destroy these latently infected cells to actually achieve a cure? So I'd say one of the key hurdles for the HIV cure field is that we typically study HIV-infected cells in a vast sea of irrelevant uninfected cells. And a lot of this has to do with a very simple numbers game. If we look at, for instance, peripheral blood from an HIV-infected individual, somewhere in the order of 1 in a million cells is actually infected with HIV and is harboring HIV. And since we typically take large volumes of blood and then we look at average behaviors within those cells, really, the biology of the latently infected cell is barely even represented in that mathematical average. So I think the field is changing dramatically in the last few years. The advent of single-cell technologies is totally changing the ballgame. And our lab in particular is pursuing 3 different technologies in collaboration with other groups. So first, we're doing a lot of single-cell RNA sequencing or transcriptomics. And this is a collaboration with the Bio-Rad digital biology group. We're doing something called single-cell attack sequencing. So attack stands for assay for transposase accessible chromatin sequencing, I think. And so the idea behind attack-seq is we can come up with a comprehensive map of the chromatin landscape across the entire HIV genome to see which regions of the genome are accessible and which regions are not accessible, for instance, to the transcriptional machinery. So I kind of think of it as complementary to single-cell RNA sequencing, except we're looking at things at a much more macro genome-wide scale. And then lastly, we're doing single-cell DNA analysis, so we're doing targeted sequencing of the genome in infected cells in collaboration with a local startup called Mission Bio. So first, to talk about the profiling part, I'm going to introduce you to this ddSEQ single-cell transcriptomics platform. And so all of the single-cell work in our lab is really championed by Dr. Konstantinos Georgiou, who is a postdoctoral fellow in the lab, who also happens to be a current participant in the CFAR mentoring program. And so the way this technology works is each individual cell is encapsulated in a single droplet. And at the same time, coming from the bottom, or coming from the top, you have beads that are coded in barcodes, and those barcodes wind up in the same exact droplet. Then in the downstream sequencing interaction, if you deconvolute all those barcodes, you can link particular transcript sequences to particular individual cells that were in your original sample. So this is what Konstantinos has been doing to apply this single-cell sequencing approach to study HIV latency. So first we start with the HIV duofluo virus, and I'm guessing some of you are familiar with this virus. This was a dual reporter virus that was originally created by Eric Verdon's group. And as you can see here, it's got a green GFP reporter that's driven by the viral LTR that reports when the virus is transcriptionally active. And then it's got an MKO2 reporter that's connected to an EF1 alpha constitutive reporter. So long story short, when the virus integrates into the host genome, this MKO2 reporter lights up. And then if the virus also happens to be transcriptionally active, then the virus is dual positive. You'll also get a green signal. So Konstantinos takes these cells, and this is an example of what the reporter infected cells look like. So cells here that are GFP positive, that are MKO2 positive, are infected with HIV, but they're not producing any viral transcripts or proteins. And cells up here are infected, and they're also actively producing viral components. So he uses flow cytometry to sort those cells, and then he loads them into this ddSEQ single-cell sequencing platform. And then we use the Illumina NextSeq sequencer to sequence those droplets, and then we do data analysis. and to expand the data analysis, there are multiple bioinformatics steps associated with this process. The first step is we take all of these sequencing reads and we align them against both the human genome as well as the HIV reference genome. So in addition to characterizing human gene expression, we're also interested in characterizing patterns of viral gene expression. Then we do a range of secondary analyses, including the application of machine learning algorithms to decipher a signature associated with latently infected cells. And then really where we're going with this is we're hoping that this approach will yield new pharmacological targets, so new targets that we can dial into to manipulate this process and target latently infected cells. And equally importantly, we're hoping that this approach also yields new biomarkers that we can use to measure the size of the latent reservoir, which is a real key deliverable that the HIV cure field needs to monitor its curative interventions. So the first thing we do is we remove confounders, and I'll explain this very simply. As you can see in the top, there's a strong donor effect in the data. So the distance between each one of these dots, which represents a single cell, reflects how closely related they are. So you can see clearly that different donors are clustering by themselves, suggesting that they have a distinct signature that's unrelated to HIV infection. So we use a series of bioinformatic methods to strip that variability out of the data, and then you can see that everything regresses into one kind of coalescent blob. So if we apply this approach called TSNE, which stands for T-distributed stochastic neighbor embedding, then we can start taking a look across donors to see if there are any signature patterns associated with latently infected cells as opposed to uninfected cells or productively infected cells. And as you can see by the color coding in this diagram of the different cellular populations, all of the latently infected cells, which are labeled in green, tend to cluster together. So after we delete a lot of this variability that's just a signature of the different donor, then we can actually see patterns in the data that suggest that latently infected cells do in fact share gene expression programs that are completely distinct from productively infected cells, uninfected cells, and also cells that have not been activated. So then we apply both a conventional statistical approach to identify genes that are associated with HIV latency, and then we've also been using a few different machine learning tools to explore the transcriptome of latently infected cells. And what we can see here is that a very simple decision tree involving just 4 genes can very accurately classify latently infected cells and distinguish between latently infected cells and both productive cells and uninfected cells. And since I don't have the time to really explain the mathematical methodology, I can just tell you that this is a very statistically conservative approach. We're using an approach called cross-validation, where we only train on a subset of the data and we test on the remaining portion of the data. And even using that rigorous approach, we're able to achieve a specificity of 92%. So we rarely call a cell as a latently infected cell if it is not a latently infected cell. And this volcano plot shows that there are a range of factors, there are a range of genes that are overexpressed in latently infected cells with respect to productively infected cells. And there are also a few candidate genes that are very significantly underexpressed with respect to productively infected cells. So I think what the field really wants, and there have been a couple of very prominent papers on this in the last couple of years, the field really wants a surface marker of HIV latently infected cells, because even if we're lucky enough to find 1 or 2 mRNA transcripts in a cell that universally label HIV-infected cells, it's pretty hard to go inside an infected individual and find a cell based on a transcript inside it. So we really need to find proteins on the surface of those cells. So what Konstantinos did is he leveraged this database where they've taken essentially the entire family of human transcripts, and they've classified them into transcripts that encode surface-expressed proteins versus transcripts that encode proteins that stay within the cell. And when we focus our search specifically to factors that are predicted to be on the surface of latent cells, once again, we actually find in our data that there's a very small group of surface-expressed targets that very rigorously segregate cells between latently infected cells and then productively infected and uninfected cells. And intriguingly, almost all of the targets that we find in this search are biologically intuitive factors. For instance, ICAM3, which is associated with extracellular signaling that is associated with the transactivation of the virus. So something I don't have the time to talk to you about today is the fact that we're complementing all of this technology with another approach called nanostring digital barcoding technology. And here we're skipping past the guesswork of predicting the surfaceome of infected cells. We're actually using an approach that can both measure proteins and transcripts at the same exact time. And so Hannah has been using this tool to study latently infected cells ex vivo as well as cells infected in these different latency model systems. And she's been able to identify a couple of surface proteins that very reliably segregate with latently infected cells. And since I don't have time to present this today, Hannah is presenting all of this in a talk at the Gladstone Institute next Thursday at 3 p.m., and her talk is called Encountering HIV Persistence Using Nanostring Encounter Technology, and she can definitely do a much better job at describing her data than I can. So the conclusions on the profiling part of the talk. Latently infected cells are characterized by a unique expression signature, including RPS6, TPT1, SLA, and then TNF-associated factor TNF-AIP3. The surfaceome signature of latently infected cells is likely complex. So I don't think anything in our data support the idea that there's one silver bullet that's going to be a marker that's always on infected cells and it's not on anything else. I think that would be ideal if it was the case, but what we're really seeing is that there's probably a complex array of a handful of molecules that allow us to classify latently infected cells rather than one silver bullet. And lastly, just a methodological statement, single cell analysis coupled with machine learning tools, I think it really represents a very powerful knowledge discovery approach that will be useful to the HIV cure field. So work in progress and future directions on this front. In addition to the single-cell transcriptomics work I mentioned, Konstantinos is pursuing the single-cell attack sequencing approach to look at the chromatin landscape across the entire genome. And right now we're in the process of setting some confirmatory experiments where we can use CRISPR-Cas9 to manipulate some of the targets that we're finding in these screens to see if they actually play a mechanistic role in the establishment of latency. And at the same time, we're also collecting samples from the scope and options cohorts and then running profiling tools on those samples to see if any of the factors that we're identifying in our ex vivo search are actually recapitulated in vivo. So very quickly, I'm just going to skim through what we're doing on the purge side of the equation. I already talked about profiling. These are some of the things that we're doing to actually purge the HIV reservoir. So I'm sure everybody in the room is familiar with the whole CRISPR-Cas9 revolution. And for anybody who's ever tried to edit a gene in the lab, there is no doubt that the discovery of the whole CRISPR-Cas9 system has completely revolutionized the way that we can edit genes. So beforehand, actually delivering some sort of genetic modifying agent to a particular gene was incredibly difficult. But now it's been made very simple. And so we're very aggressively exploiting the CRISPR-Cas9 system to develop a bunch of different cure approaches in the lab. And the people who are really driving this work are Leonard Chavez, who's a staff scientist in the lab, who's also a former CFAR mentoring program participant, Kyle Raymond, who is really Leonard's second in command, who's doing really amazing molecular work, and then Mohammed Bouzidi, who rather than using lentiviral vectors, Mohammed is using the Cas9 RNP system. And a big shout out to members of the Alex Marson lab, including Ujwal and Joe Hyatt, who have really helped our lab bring some of these tools into our own space. So very simply, how the CRISPR-Cas9 gene editing system works, basically you can just take a string of nucleic acids, you can take something called a guide RNA, which is homologous to a particular gene that you're interested in editing, and then you can use it to recruit this Cas9 gene editing enzyme to a particular site in the genome. And so again, it really takes a lot of the guesswork out of the equation, because as long as you know the sequence of the gene that you're trying to target, you can pretty easily program the Cas9 protein to wind up at that site and cut it. And so what really happens after the Cas9 enzyme arrives at your gene of interest and cuts it, the non-homologous end joining machinery within the cell tries to rectify the mistake. So it tries to reassemble the DNA back together. But when it does that, it usually does not do that perfectly. So some fraction of the time, the wild-type gene will be preserved, and some fraction of the time, there'll be either an insertion or a deletion that's introduced into the gene, so you no longer have the translation of a functional protein. So there are two basic ways that we can use this technology. One is we can edit cells ex vivo, and then we can reinfuse them back into an individual. Or we can actually deliver an editing vehicle into an individual and actually have the gene editing occur inside the infected individual. And within the scope of HIV cure research, I think there are two basic ways that we can use the technology. With the ex vivo editing, we can take cells out of an individual, and then we can genetically reprogram them so that they're resistant to HIV, and this has been going on now for at least a decade. We can also create super potent anti-HIV immune cells and then put them back inside the individual. In terms of in vivo editing, and this is something we're very interested in, we can directly target the HIV provirus in infected cells by sending in essentially a giant pair of molecular scissors to go into every infected cell and cut the virus in place. But there's some very serious obstacles facing the idea of using CRISPR-Cas9 in the clinic at all. One of the main obstacles and concerns is this idea of off-target editing. Even though we're intending for Cas9 to go to one particular gene and cut it, there's some non-zero probability that it'll go to another locus and it'll interfere with that, and that could have all sorts of undesirable consequences. The other thing is viral diversity. So HIV is incredibly genetically diverse, and as you can see in this slide, the size of the phylogenetic tree represents global circulating HIV diversity. So you have this massive blob here. If you look at this blob to the right, that represents the entire worldwide influenza genetic diversity, which is several orders of magnitude smaller, and we still have trouble designing an effective vaccine against that. So just like viral diversity gives us issues with vaccine design, it's definitely presenting a serious challenge in terms of gene editing. And because of this diversity, we also have the evolution of escape mutants. So Leonard has championed a series of approaches to clear all of those hurdles, and I definitely don't have time to describe them to you in detail, but I'll just give you a real quick aerial view. So the first thing that we did is we contacted my former mentor, Dr. Betty Korber at Los Alamos, and we took her vaccine design algorithm. So she's designed this amazing set of algorithms to design HIV vaccine antigens that have maximal coverage across the entire HIV-M group. We repurposed those algorithms to identify maximally conserved guide RNA targets within the HIV-M group across the HIV genome. After we did that, we did some very exhaustive testing of these guide RNAs in vitro to figure out which ones actually had activity against the virus. And then we came up with a list of about 6 or 7 top candidates that are extremely well conserved that also do a great job at cutting the virus up ex vivo. And then Leonard and Kyle have worked together to engineer a family, really, of therapeutic lentiviral vectors that express Cas9 and also package multiple guide RNAs in a single vector. And they're now doing a whole bunch of experiments, both in Petri dishes and also in a mouse model, to really come up with a very fine-scale estimate of how potently this construct can edit HIV in vivo now. And lastly, we've paired with this company, Mission Bio, to look at the CRISPR-Cas9 gene editing on a single cell level. So to explain the problem, even if we can deliver this vector into cells or even into people, it's very difficult to understand how efficiently this technology is working without really looking at single cells to see how many of the viruses are being genetically perturbed. And this technology that's produced by this local Bay Area startup has really given us the capacity to interrogate the genome of single cells. The other thing that we're trying, besides the lentiviral approach, we're actually working with Padmini Pillai, who of course looks exactly like me, who happens to be my kid sister, who is a postdoctoral fellow at MIT in the Bob Langer lab. And she's doing all of her research on developing lipid nanoparticles to deliver vaccine antigens. And actually, she just recently started working with Bruce Walker and others at the Reagan Institute to package some of their vaccine antigens into nanoparticles to test them in vivo. So what we've been doing with my sister is we've been sending her guide RNAs, and then she takes them and packages them into her nanoparticles and then sends them back to us. We are really a nerdy family, extremely nerdy family. I didn't really think about that until I said that, right? We are a nerdy family. But we also do stuff like this where we break lots of laundry baskets, and we've been doing it since like 1984 or something like that. So I also wanted to say all of this stuff, which I glossed over very quickly, Leonard is going to give one of the main featured talks at Bay Viro, as is Nadia and some of the other people in our collaborative universe. The Bay Viro Symposium at UC Berkeley is amazing. This is the 9th one this year, and I highly recommend that people here attend the symposium next Friday at UC Berkeley if you have a chance. So I'm just going to close with this one slide. The other thing that we're also experimenting with on the purging front is this idea of taking exosomes or extracellular vehicles and programming them to use them as agents to target and destroy HIV latently infected cells. And this is work conducted by Pooja Bharadwaj, who is a current participant in the CFAR mentoring program. And Pooja is working with this company called System Biosciences that focuses on exosomes. So exosomes are these membrane-bound vesicles that all of our cells naturally produce. The idea is that they're generated to facilitate intercellular communication. And what Pooja is doing is she's taking these exosomes and she's repurposing them to use them as delivery vehicles. And this approach has already been used successfully to some extent in the oncology space. And so we're kind of trying to mirror that now in an antiviral setting. But so what she's doing is she's taking exosomes and then she's using this technology developed by this System Biosciences company to label the surface of the exosomes with anti-HIV antibodies. And then she's taking HIV-infected cells and incubating them with these designer exosomes. And what we've been able to show and what she'll demonstrate in her talk next week is that we can both use these exosomes to block HIV infection, and we can also use them to selectively identify HIV-infected cells, with one huge caveat, providing that they're expressing HIV envelope at the cell surface, which is of course going to be a huge challenge for anything like broadly neutralizing antibodies where we need the expression of envelope. But the hope is an approach like this could be coupled with like an HIV latency reversal approach in the future. And since I'm plugging the entire lab, next week there's also a great symposium on Thursday right next door at Genentech Hall called ExoFest, which is an all-day symposium just focusing on exosomes, both on using them as biomarkers of pathogenesis and also to use them as designer drugs, essentially. And Pooja will be presenting all of her research on reprogramming exosomes at the ExoFest next week. So I'm going to close with my acknowledgements here, just to make it very clear. This is certainly not a comprehensive list. I don't even know how to make it clear, but I've been a complete product of the CFAR system. I trained at the UCSD CFAR with Doug Richmond and also with Joe when he was down there. Had a wonderful experience there. Then I came here, and I've been treated so well here, really. And Paul and Warner have been absolutely wonderful. It's just been great. But I also want to thank all of the participants that provide clinical samples to us. And I'm going to get teared up here right here, but I also want to thank our foosball table, which is really the centerpiece of our entire lab operation. All right, thanks a lot.
Questions, comments?
Not a lot, huh?
Oh! Who's got your phone? There, Deanna. Dr. Finzi.
Great talk. The CRISPR question, so there are several hurdles, but I'm wondering about the hurdle once, even if you narrowed it down to a certain cell, if Nadia and you together narrowed it down to getting the CRISPR in, but then you have to get it into the cell, into the nucleus, and then finding that DNA that could be anywhere and can be completely closed
Sure.
up in the. So.
How do we do that?
How does?
No clue. No, okay. I'm going to come up with something. I'm spitballing here, but no. So I think there's two philosophies here. So I think you're 100% right that a lot of the reason why a lot of these viruses are latent is because they're so deeply repressed in chromatin that they never see the light of day. And so I think that Cas9 probably would have almost no chance of actually winding up, finding those insertions. So I think one possibility, and I don't think this is really backed by data, but there's it's intuitive, I think. One possibility is some of the most deeply latent viruses are maybe ones that we don't need to care about because the possibility that they'll reactivate in the near term is not so high. That would be convenient, but I don't think we can bank on that possibility. So I think what we'll have to do, and I think the attack sequencing approach that Konstantinos is using will be really informative. I think we need to figure out if viruses are very deeply repressed in chromatin structure. I think we need to figure out how we can use agents to remodel the chromatin structure within the infected cells to release the virus, at least temporarily. And this really interfaces directly with the shock and kill idea. I'm not sure it'll be the exact same set of tools, but I think the idea of activating the cell in some way to release chromatin structure to make the virus visible will be a critical part of that conversation.
Yeah, thank you. That's exactly my question. So CRISPR doesn't work so well if the chromatin is all, or is it not known?
So here's my thing. I think that's the explanation. I've never seen a publication that actually says chromatin state is a determinant of CRISPR accessibility. I think it's very intuitive that it is, and I think there's a lot of circumstantial evidence that suggests that. So if you take primary cells and then you try to genetically modify a bunch of targets, and you don't activate the cells, you can hit some of them. So some of the genes will get edited, but a lot of the genes are not editable. As soon as you activate the cells, like for instance, if you take a T cell and you blast them with CD3, CD28 stimulation, all of a sudden your editing efficiency goes through the roof. And I think the simplest explanation of those data is that you're essentially releasing a lot of those genes from deep chromatin repression. And I think the same exact mechanism would apply to the provirus as well.
Thank you.
Thank you.
Satish, this might just reflect my obviously limited intelligence, but you think of truly latently infected cells as not producing any antigen. So how is the surface of the cell going to, why would we think that there's going to be a marker of latency on the surface of a cell?
Excellent question. So if the virus is really completely just doing nothing, why should there be a signature? So two things. One is, I don't think we really know that nothing's happening in those cells with the virus. Like we always, I think people, there's almost kind of like this historical artifact where people have completely equated the transcriptional latency of the virus with latent infection. I don't think that's necessarily true. I think a latently infected cell is a cell that's harboring a replication-competent virus that's somehow figured out how to hang out for a long time. And one possible way that's happened is the virus isn't really doing much, so it's not dying of cytopathic effects. So I think there still might be something brewing with the virus. But even if we assume that what you said is true and the virus is doing absolutely nothing, I think that there's this idea of historical chronological recording that happens in the cell. And one thing that happens with HIV is that you actually have integration into the host genome. So at some point, the virus entered that cell and then it cut the DNA and entered it, unless if it was through clonal expansion. But if it was actually infected de novo, there was a double-stranded break in there, and then the virus had to repair that. And I think it's possible, and we've seen bits and pieces of hints in our data actually, that there's a record of that insult of the original infection, and that might persist even after the virus stops doing anything.
Great. Another question?
Hi, Patricia.
There's a microphone coming to you.
Thank you, Satish. Always a pleasure to hear you. And you forgot to say what a talented musician you are.
Oh, yeah, that's right. Excuse me. I'm a very talented musician. All right, now I got that out of the way.
Well, question. You're going to hate me for this question, but, in FAR we talked about the cure for 2020. So I wonder, how far do you think we are for an actual cure or something that we can actually move into trials?
Well, we're already moving into trials, right? I think there's enough promising things happening where we're already moving into trials. I don't think we're really moving into trials with anything in our current lineup where we really think that we're going to cure 100% of the people that get treated with anything. I think there's lots of promising data coming down the pike with broadly neutralizing antibodies and a lot of different approaches where I think they're kind of coalescing into tools that we can develop. I think a lot of the answer to your question also depends on whether you're talking about complete eradication or this idea of a durable remission. I think complete eradication I don't think it's impossible, but it's just such a lofty goal. I think the idea of sort of mimicking the whole cancer treatment space and figuring out something to have a durable remission that's durable enough to be meanful, where people can go off of small molecule drugs for several years on end. I think that's possible, and I think that's something that we'll see in the not so distant future, but I'm not much of a gambler, so I can't tell, but I'm optimistic. Oh yeah, and I remember even when I came to your the Comhar thing with Nadia this question was brewing all over the place. It's a very obvious question, but I think I can guess, but I don't really know. But I'm optimistic.
Hi, Satish. I just have a really quick
Susanna.
question. It's really beautiful work.
Thank you.
Just in regards to your transcriptomic data, with the duofluovirus, you find that, I think it's really nice, the cells cluster as per you would expect with the productive and latently infected. But what I noticed was it looked like the productive cells were clustering more closely to the
Uninfected? That's right.
and the latent sort of formed a distinct population. Do you have any insight into why that might be and what transcriptional factors might be involved?
So this is something we're really excited about. So both HANA's nanostring data and all of the single-cell data show us the same thing. So we always just expected by default that the uninfected cells would look most closely related to the latent cells, and the productive cells would be over there because they're, cranking out viral transcripts, and so they're going to be grossly different. Nope, everything that we've done shows the exact opposite, that productively infected cells more closely mirror the uninfected cells, and the latent cells are off by themselves, which is really awesome, actually. I think that really gives us hope that there is a true signature associated with them. In terms of the exact factors that are driving the difference, I think the list that we've developed, that I didn't really go into individual genes, I think those factors that we've identified really tell us what we need to know. And I won't go through this now, but you and I can meet with Hannah and she can show you, but I'll just show you this one thing real quick. This cool flower right here, this flower actually is like this, Venn diagram on steroids that actually has pinpointed the exact factors that are responsible for that clustering pattern. So we could pick Hannah's brain after this, I think. Thanks.
Great, thank you. Thank you, Satish.